Okay, well, let's go and get started. So the only announcement would be, I think, pretty obvious. On Wednesday, we're having an exam. So be ready for that. We won't have any assignments due until next week, besides that. Okay, working on your problem sets right now in terms of getting them graded. Hopefully by Wednesday, you'll see grades for those. Okay, I would like to say, talk to one of my TAs in regards to the problem set. He said scores were really good for the stuff that he had graded thus far. He said that it did seem like people were a little bit confused on the fact that electrons have to be recycled. There has to be a recycling of them through the Q cycle. Okay, just remember that every 2 electrons that enter into complex 3, one of them is going to turn right back around and get back on a ubiquinol molecule. One's going to go through. Okay, so just keep that in mind, especially as you approach the exam. Okay. All right, so our riddle here, complete the sequence of letters. What is it? E and T. Very good. 8, 9, 10. Right? The first letters of 8, 9, and 10. Because these are the first letters of 1, 2, 3, 4, 5, 6, 7. 8, 9, 10. Okay. It looked tricky, and then it really isn't. It's just counting. Okay. All right. So we have a little bit of material to finish off our current lecture, Lecture 9, just one slide really, and then we'll go ahead and jump into Lecture 10. I really need to keep going. We're behind. I don't want to get too far behind, otherwise we won't be able to cover all the material needed for this class in Exam 4. This is Lecture 9 though, and at this point in time, we have on Friday discussed the mechanism of how ATP synthase can use a proton gradient to make ATP. The proton gradient is driving conformational changes. Those conformational changes then lead to essentially ejection of ATP from the active site. Of course, the conformational changes are at that C ring in the A subunit that cause that C ring to turn. Okay, and when it turns, then it turns the gamma shaft, and that then causes conformational changes in alpha and beta. Okay, keep in mind that the reason that protons are going to move from P to inside is because there's a high concentration of protons on the P side and low concentration on the inside. Okay, and that then favors in the A subunit, the half channel of the A subunit favors, that's facing the P side, it favors protonation events. Okay, that will then allow for that aspartate or the glutamate to be protonated, causing a conformational change. On the inside half channel, there's low proton concentration, which favors deprotonation, which will also cause conformational changes. Okay. Now, we're looking at here a diagram of an experiment that was done. So everything that I have shown you up to this point in time has been a cartoon, and hopefully that cartoon was helpful in allowing you to understand how ATP synthase works. However, it is just a cartoon, and it would be nice to look at some actual data that support that model that I illustrated or showed you. And so what I want to do is talk a little bit about this experiment that was actually published in one of the premier journals of in science, and that is actually the journal Science. And this experiment is really cool. Okay, so this research group, what they did and what their hypothesis was is that if the production of ATP by alpha and beta require a clockwise change in the C ring, a clockwise turning of the C ring, then alpha and beta, if they were to hydrolyze ATP, then they would cause the exact opposite turn in the C ring. Okay, all enzymes can catalyze reactions in the reverse direction, reverse of what we see in a cell. And so it's very possible then that you can take these alpha and beta subunits, give them ATP, and have them, if conditions are right, hydrolyze ATP. That energy can be then transmitted to actually turning the C ring instead of the C ring turning, creating ATP, right? And the reason they wanted to do this is because in an experimental setting, it's very difficult to set up proton gradients. Okay, because you require membranes and intact membranes, and that's a very difficult thing to do. This is a lot more elegant and a lot simpler to do. So what they did is they genetically engineered an ATP synthase molecule, and they genetically engineered it, first of all, on these alpha subunits to have what are considered his tags. His tags are just a string of 6 histidines in a row. Okay, and his tags will actually have a pretty high affinity for nickel. And so what you see here, this here, that plate is a nickel plate. And so by putting these his tags on the alpha subunits, they could properly orient any ATP synthase molecule that they had expressed in their expression system, so that when they looked at the nickel plate, they knew that they were looking down, okay, on ATP synthase. In other words, their eyeball is going to be right here, okay, and they could ensure that all of them are oriented in one direction. Now, the other aspect of their genetic modification was this actin filament, this big thing that just doesn't belong there on the C ring. Okay, and an actin filament is found in your muscle fibers. Actin is in a monomeric form is a protein that actually can polymerize and form these insoluble fibers. It's a filamentous type protein. And so they then engineered an actin filament to be on the one of these C subunits, or one of the C subunits of the C ring. Now, the great thing about actin is that it will selectively absorb fluorescent dyes that most proteins won't. So you can fluorescently dye an actin filament, and then if you have a fluorescent microscope, you can observe the actin filament. Now, having that, then, what they have is a system where they can orient the ATP synthase molecule, give it ATP, and using a fluorescent microscope can see if ATP hydrolysis will lead to the movement of that actin filament. And I wouldn't be telling you about it, and it certainly wouldn't have been published if it didn't work, and it did work. It's really cool. So, so what you're looking at here, the arrow is pointing to it, it's really small, but that is one of these actin filaments that's connected to the C ring. Okay, and they're going to give ATP to this, and you'll watch it then start to turn in the counterclockwise direction. Oh, see, it's kind of jumpy for you guys, but that's okay. You can kind of get the idea that it's moving in this counterclockwise direction. That's so cool. That is really, really cool. Okay, so that was direct evidence that C ring movement is related to, in this case, ATP hydrolysis, but it could go in the opposite direction. Okay, all right. So very cool stuff. Questions on that? I didn't hear anyone say, ooh. Yeah.
Is it the same efficiency in reverse as this forward or close to it?
So I don't know. So the question is it the same efficiency in the reverse or close to it as in the forward direction? My presumption would be yes. So they have actually made, and I don't know the exact numbers on this, but they've actually made measures of efficiency, because you can actually calculate then using the system the drag on the actin filament, and you can calculate the amount of torque that would be required to put that kind of drag on this, and the rate, and all sorts of things. So they've done some really interesting experiments with this setup. And they've demonstrated that the ATP synthase molecule, and I don't have percentages, but it's actually really efficient in terms of the use of the energy to make or use ATP. Okay. And of course, you're making it from, you've got a proton gradient, electrochemical gradient that's being converted to mechanical energy that then is being converted back to ATP, or chemical energy. So in this case, it would be chemical energy being converted to mechanical energy. So, yeah. Okay. Other questions? So cool. So cool. Okay, that's it for lecture 9 then. Okay, that's all we need to say. Now, I guess one more thing to say here. The ATP synthase molecule actually can be used to hydrolyze ATP in the cell. It can establish a proton gradient. Instead of using a proton gradient, it can make a proton gradient using hydrolysis of ATP rather than making ATP. Okay, so some interesting things about this molecule, and this whole chemiosmotic theory and molecules like this, there's been lots of them that have been discovered that do similar things. Flagella movement actually in bacteria is driven off of similar ideas. A lot more complex model though. Question, yeah.
So is the step that's actually getting the energy the binding of ATP into the pocket, or is it the hydrolysis step?
Oh, okay, yeah, interesting question. The question is it the actual hydrolysis step, or is it actually the binding of ATP that causes the conformational change? I don't know the answer to that question. My presumption would be actually that it would be, well, you'd think that it would be the binding based off of what we talked about if that reaction coordinate diagram that we looked at earlier is correct. That the binding there, the binding energy would be translated to the conformational change. But I'm not entirely sure. Yeah. Question?
Is it known, like how much, like as far as the ATPase, how much of a protein or proton gradient it actually needs to function, like the minimum for it versus?
So I don't know the minimum that it needs to function. I do know that generally speaking, the difference in pH between the two compartments, N and P side, is somewhere around 0.75 pH units. You don't really see it exceeding that, but that would be more of the thermodynamics and electron transport chain. But I don't know if it is their minimum. It would be obviously, at conditions prevailing in mitochondria, you're going to need about 40 to 50 kilojoules of energy to produce a single mole of ATP. Okay, if you presume that you still have about 21 kilojoules of energy in one proton, you probably do a calculation backwards in terms of how much of a gradient would you need to do that. But I'm not sure. So.
The reason why I ask is, like, I kind of wonder, like, how close or I should say, if there's any wiggle room as far as the electron transport chain, like how much of a proton gradient it can produce versus how much is required to actually drive this, if it's possible to engineer it.
Yeah, I don't think that you need to have a maxed out 0.75 pH difference between the two environments. I think it would depend. I think there's factors that you have to consider in terms of how big the C ring is as well. Because the energy stored in the proton gradient is entirely dependent upon the difference in that pH, right? I talk about 20 kilojoules of energy in a mole of protons moving from P to inside. There's about 20 kilojoules of energy available in that. That number is dependent upon the fact that the difference in pH would be 0.75 units between the two environments and a membrane potential of about 0.15 volts. Again, I don't know the answer if there's a minimum, but I'm sure there is a minimum. I mean, obviously it can't be at equilibrium, but I don't know what the minimum is to get it to start to turn. So, okay. Other questions? Okay, awesome. Well, let's do this. Let's shift gears. So that finishes the material that will be on Wednesday's exam. And let me go ahead and start some new material. This will not be on this upcoming exam, but of course the fourth exam in the class. Okay, our last exam. So we're shifting gears once again. We shift gears from carbohydrate metabolism into cellular respiration. And now we're going to shift gears from cellular respiration into lipid metabolism. There are actually going to be two different sets of lecture notes on lipid metabolism. This first one we'll use this as an introductory slide, an introduction to lipid metabolism. That's what we'll do primarily today. And introduction to lipids, generally speaking. And then we'll talk mostly in this lecture about how lipids are transported through our body. There are some very intriguing challenges that have to be overcome when you start to think about lipids moving through our body, because lipids are primarily hydrophobic. and they have to be transported in our bloodstream, which is primarily an aqueous environment. So we'll talk a little bit about that. We'll actually talk quite a bit about that. And that actually transport is directly related to lots of different cardiovascular diseases, at least abnormalities in it. So we'll visit some of those topics as well. Okay. So, and then in our next lecture, on lecture 11 on lipid metabolism, we'll actually look at the pathways that break down lipids and make them. Okay. All right. So let's do this though. Let's consider an introduction here. Like I said, shifting gears, we have a new carbon source for energy production, and I want to go ahead and compare and contrast carbohydrates to lipids. It turns out that that lipids, you can store a lot more energy in lipids than you can in carbohydrates, and there's actually two primary reasons for that. The first one is due to the level of the redox state of each of the carbons in the lipid. Okay, so let me go ahead and draw a carbohydrate first. So we'll just draw glucose here. 6 carbons. Each little spot that a hydroxyl group's coming off of that line is a carbon, and then of course the 2 carbons at the end. So this is a 6-carbon molecule. Okay. If you were to take just strictly dry weight of a carbohydrate, there is about 17 kilojoules of energy per gram of dry weight of carbohydrates. Okay. Depends on the carbohydrate, but that's on average. Okay, about 17 kilojoules per gram. Compare that to a lipid. Draw a lipid over here. This will be palmitate, or palmititic acid. We could draw lots of different ones. Okay, it depends on it would just change the length of the chain. These, though, will have about 38 kilojoules per gram, okay, of energy. Right? So 38 kilojoules of energy per gram of lipid can be found in lipids. Almost a little bit more than twice as much energy just in dry weight of the carbons in a lipid versus those of a carbohydrate. And the reason is this, is the redox state, right? So this carbon here, okay, is a more oxidized version of a carbon because it's bound to oxygen than, let's say, one of these carbons where it's only bound to hydrogen. Okay, so this is a more reduced, okay, more reduced state of carbon. So when we start to think about this from a metabolic standpoint, as electrons are taken off of lipids, it's going to lead to higher productions of NADH and FADH2 that can feed the electron transport chain. Of course, that's going to lead to more ATP. Okay, so you can store more energy in a lipid just straightforward from the carbon standpoint. They're more reduced than they are in a carbohydrate. Okay. Now the other aspect of this has to do with their insolubility. Okay, so these are insoluble. In water. Okay. Now that can be an advantage and it can be a disadvantage. There's two aspects of this. We'll think about the advantage first. Okay. The advantage of this is that they can be stored anhydrously, okay, without water. Whereas a carbohydrate, you have to store, when we store carbohydrates or energy as carbohydrates, it has to be hydrated. There'll be a whole lot of water that's surrounding it as well. Okay, it's completely soluble. Okay, so when we start to look at this, these are the values I gave you here. Those are dry weights. Okay, but in a biological context, because water will solubilize the carbohydrate but not the lipid, there is actually, okay, lipids store 6.75 times as much energy compared to glycogen. Of course, glycogen is the means of storing energy as carbohydrates. Okay. So it's twice as much energy when you look at the dry weight, but it's about 7-fold more energy when you look at it actually solubilized in a cell, because the water adds significant mass to glycogen where it doesn't add anything to the lipid. Okay. Now here's the importance of this. Okay, so if we were to consider, okay, a 70 kilogram male, we'll do a male, okay. Your average 70 kilogram male, they will have about 11 kilograms of lipids storage. Okay? About, on average. That is equal to about 420,000 kilojoules of energy storage. Okay? So that is enough to fuel bodily processes, not for days, but actually weeks of time. Okay? So without any input of any external sources, right? So you can have extended fasts with that amount of energy stored on you, right? I can't remember the numbers, but your typical obese individual would have enough energy storage and lipids to survive about a year. So, you know, certainly more than what you would need. Okay. Now let's compare that, okay, situation to trying to store that much energy in carbohydrates. So if you're to try and store that much energy in carbohydrates, 420,000 kilojoules of energy in carbohydrates. Okay. So to store 420,000 kilojoules of energy in carbs, you need 64 kilograms of carbs to do this. Okay. Glycogen. Of course, 64 kilograms of carbohydrates is essentially the entire mass of the individual. Okay, which, you know, obviously we're not going to do that. Okay, so this just highlights for you how much more energy can be stored in lipids, and why actually lipids would be a preferred means of energy storage. Because number one, it's a more reduced form of carbons, and secondly, it's anhydrous. You don't have to. This weighs significantly less to store that much more energy. Okay, and so your body, when there's excess energy, it will convert that energy to fat and store it in adipose tissue. Okay, so obviously, diets high in carbohydrates lead to lipid storage, not carbohydrate storage, lipid storage. Okay. Questions on that? Fascinating stuff. That's an introduction to this. Now, this is, right, I mean, when you start to think about this, the fact that these are insoluble, there's an advantage in terms of storage, but there's also disadvantages. Okay, and we're going to think about those disadvantages, and that's primarily to do with how do we move these things around. Okay, you have hydrophobic molecules that have to be moved through aqueous environments. That poses some challenges, so we'll have to consider that. Okay. All right, now let's move on here and let's give an introduction to lipids. There are three main classes of molecules that we would consider quote-unquote lipids. Okay, the first class, actually 2 slides on this to introduce this, but the first class would be triacylglycerides. Okay. And triacylglycerides, TAGs, they can also be, if you look in the primary literature, they'll go by TGs, triglycerides, or triacylglycerides is another one, TAG. I've seen both in the primary literature. Okay. So triacylglycerides, first of all, are made up of 3 what are called acyl chains or fatty acid chains. Okay, so what you were looking at here, this would just be a fatty acid, or maybe what would be called a free fatty acid. Or FFA. Okay. Free fatty acids have these hydrocarbon chains, the tails, what would be called the tails, and then a carboxyl group, which would be a polar head group. Okay. This one here would be saturated. Okay, and hopefully this is some review for you in terms of lipids. Saturated meaning that all the carbons in the hydrocarbon chain have single bonds between them. Okay. Over here, we'd have another free fatty acid. It has a double bond in it. It produces this kink in the molecule. This would be unsaturated. We would say it's monounsaturated because there's only one double bond, and it happens to be a cis double bond. Cis double bonds are what we find in naturally occurring lipids. You don't really find trans double bonds in naturally occurring lipids. Okay. So generally speaking, cis. Okay. Those are free fatty acids. Very rarely will you find a fatty acid in its free, quote unquote, free form. Generally speaking, we will find fatty acids as acyl-CoAs, okay, where the R group here would be our hydrocarbon chain. Okay, and they'll have this coenzyme A molecule attached to them. That's how you would find quote unquote free fatty acids in a cellular environment, is an acyl-CoA. Now, I put next to this, because the terminology is so similar, I put next to this the molecule acetyl-CoA. Acetyl-CoA is a 2-carbon molecule attached to the coenzyme A. Acyl-CoA can have 24 carbons attached to the CoA, okay? Or 16 carbons, or 4 carbons, or something else. Acetyl-CoA would be one aspect, or one of many acyl-CoAs. Okay, so it's a subclass of acyl-CoAs. But generally speaking, anytime a free fatty acid is generated in a cellular environment, it'll be converted directly to acyl-CoA. We'll talk about the enzymes that do that when we get to more detailed aspects of lipid metabolism. Okay. Now, that's not really triacylglycerides. You're not looking at anything as a triacylglyceride yet. Okay, triacylglycerides, we take that free fatty acid, okay, and we attach it to a glycerol backbone. Okay, so glycerol here, 3-carbon molecule. Of course, it's got our 3 hydroxides coming off of that 3-carbon molecule. That's glycerol. Okay, take that glycerol backbone here and attach to it 3 different chains. And you have then here triacylglyceride. Okay. 3 acyl chains attached to a glyceride backbone. Okay. Chains are typically 16 to 24 carbons long in naturally occurring triacylglycerides. The chain identity can be the same. And if they're the same, that would be a simple triacylglyceride, or they can be different. And that will lead to a mixed, what's called a mixed triacylglyceride. And what you're looking at over here is a mixed triacylglyceride because your chains are of different identities. Okay. Both naturally occur in mammalian systems. When we think of fats, okay, in terms of storage, this is how we store them. In adipose tissue, the fat droplet in adipose tissue is this. It's comprised of these. Okay. When we consider typically ingestion of lipids, this is the primary source of our lipid ingestion, okay, is triacylglycerides as well. And sometimes these will just be called, because of their role in storage, they'll be called storage lipids. Okay. And in terms of ingestion, I think I recall reading somewhere that it's about 85% of our dietary lipids is triacylglycerides. Okay, so it's a lot of it. Okay. It's more than 50%, let's just say that. Okay. All right, so that's the first class, lipids. Okay, second class of lipids would be phospholipids. These, certainly, we have these in our diet, but these would be considered membrane lipids. And these would be our second class, okay, phospholipids. Sphingolipids would be a part of the phospholipid family, okay. They play important roles in membranes. Let's go ahead and look at this, though. This would be an illustration of what our phospholipid would look like. Come back here. This is our glycerol. Again, backbone. We have two chains, and those chains can be different than what you have here. They can be unsaturated, they can be saturated, they can be different lengths. Depends on the membrane, depends on the composition of that membrane. Okay? But there would be only two of those, and then the other hydroxyl coming off the glycerol will have a phosphate group. Okay? And then coming off of that will be some head group. Okay? Phospholipids. Phospholipid bilayer. You've all heard of the phospholipid bilayer, right? It makes up our membrane, right? This is what that is, okay? I don't want to spend a ton of time looking at this, but we can have then X, that head group, okay, can be a variety of different things. It can be as simple as this hydrogen atom. We call that phosphatidic acid, okay? One of the most common types of phospholipids is phosphatidylcholine, which has that little choline group attached to it. To make a connection with some previous material in this class, it can be a phosphatidylinositol 4,5-bisphosphate or 4,5,6 triphosphate. Right? So what you're looking at here, this would be PIP2. Okay, PIP2 is found in the inner leaflet of many different types of membranes. It can be converted to PIP3, remember, by insulin signaling. Okay. And so that would be the head group there, and can undergo phosphorylation. And then there's a variety of others. This is certainly not even an exhaustive list here. Okay. These will be found in our diet, but they're obviously a very important constituent in our membranes. Okay. Questions on phospholipids? Okay, last type of lipid, and then we'll start talking about transport. Cholesterol is notorious. Okay, so when most people think about cardiovascular disease, the first thing that will come to their mind is cholesterol. What we are looking at here is the molecule cholesterol, but when we think about it from a health standpoint, and cardiovascular disease and the diseases associated with that, really what we'd be better thinking about is not our molecular cholesterol, but what would be considered LDLs, which we'll look at, and HDLs. Okay, LDLs, low density lipoproteins, and HDLs, high density lipoproteins, which we'll talk about. Okay, that's what is measured when we measure cholesterol in our bloodstream. And someone will say, my cholesterol is high. Okay, what they really mean is their LDL ratio with HDL ratio is off. Okay, so we'll talk about that as we move forward. Cholesterol, though, the molecule, okay, while it gets a bad rap, is absolutely essential for life to exist, or for us to exist. Okay, cholesterol, as it's shown right here, is found in membrane and plays a very important role in membrane fluidity. Okay, we won't discuss that here because we're looking at metabolism, but it's a very important molecule. Okay, it is also the backbone of lots of different very important molecules. For instance, we won't talk again about these pathways, but cholesterol is the beginning point for the production of a number of different steroid hormones, which of course are a very important aspect of our physiology. Okay, another very important aspect of this would be the fact that cholesterol, if we make some small modifications to it, can become biosalts. Biosalts are absolutely essential component of our absorption of lipids from our intestines. Okay, bile is of course generated in the liver and can be secreted into small intestine. Incidentally, the only way to get rid of cholesterol in our body is through biosalts. About 50% of it is excreted through the feces. Okay? That's the only way you can get rid of cholesterol. There's not a breakdown pathway or a catabolic pathway to break down cholesterol. Okay? There is an anabolic pathway in virtually every single one of our cells. It has about 35 enzymatic steps. You can thank me later. We're not going to go through that entire anabolic pathway. Okay? It's complicated, to say the least. Now, the other thing that's really important for our discussion in this upcoming lecture is this. The production of what's called a cholesterol ester, or CE is how it is abbreviated. Cholesterol esters, okay, are made by taking one of those fatty acyl CoAs, or an acyl-CoA. So we've come back up to here. Acyl-CoA. Take one of those, okay, and put that acyl group on the hydroxyl of cholesterol, and you got a cholesterol ester. Okay. The enzyme that does this, and we're going to encounter it a couple of times, so I'm going to go ahead and mention it, is ACAT, or acyl-CoA cholesterol acyltransferase. I like ACAT. It's easier. Okay, so acyl-CoA cholesterol acyltransferase, ACAT. Okay. We'll talk about how acyl-CoAs and cholesterol will be used to make cholesterol esters in not only the absorption of lipids from our intestines, but also in the transport of cholesterol as well as acyl chains through our bloodstream. Okay. Right. That molecule that we're talking about, these cholesterol esters, is very hydrophobic. Okay. It's got a hydrocarbon chain, and it's got that ring system, all of which is hydrophobic. Okay. So very insoluble. Questions on this? I didn't mention this, but this would be our third class of lipids. Okay. And those are the three types of lipids that we'll be thinking about primarily in this class. Triacylglycerides, phospholipids, and then finally cholesterol. Okay. No questions? Some of you are thinking, I know this. I was like this as a student. You're like, this is not going to be on the next test. And I've got a lot of stuff in my brain right now, and you're pushing stuff that I need for the next test or for this upcoming test out of my brain. It actually doesn't work that way. It's a nice thought, though, but it's not, I don't think, exactly accurate in terms of how we store things in our brain. So, but here's what I'm going to do. Because you think that, I'm going to stop right now. Okay, I'll give you 10 minutes. Use your extra 10 minutes. Okay, we'll come back on Friday and we'll start talking about absorption and transport of lipids. Okay, so 10 minutes off for today. I will see you on Wednesday for the exam. Okay. All right. Hey, what's up?